Today's lecture has several focal points associated with it. The first one is to act as a transition from the material that we just finished covering, which is classic Mendelian or transmission genetics, that is how genetic information is passed on from one generation to the next. So we'll talk a little bit about that in today's lecture. But also it serves to refresh our memories, review the topics dealing specifically with DNA structure and RNA structure and why that's so important. And then lastly, it will transition into the first aspect of what's referred to as molecular genetics, and that is understanding at the molecular level how genetic information is passed on and how that genetic information is utilized. So again, that's what we're going to focus on in today's lecture. Now, hopefully you'll recall a little bit from our previous lecture material at the beginning of the semester when we talked about macromolecules. And macromolecules, nucleic acids, fall into the category of being considered a macromolecule. DNA and RNA are both the two categories of nucleic acids, and DNA functions as the inherited directions for a cell or an organism to function. DNA is the genetic material. It contains all of the information or the instructions necessary for a cell or an organism to survive. So what we'll be doing in today's lecture is, again, hopefully getting a good understanding of how that genetic material translates into instructions or information for the cell to be able to function. In this figure, what you're seeing is a really simple diagram or schematic of DNA as the genetic material. In most circumstances, DNA is copied into a molecule known as RNA. RNA is the copy of a given gene. And in most circumstances, RNA can act as instructions to synthesize a protein. Now, when we talk about genes, there are two broad categories of genes. There are those genes that are considered protein-coding genes, and there are non-protein coding genes. The genes are found within the DNA, and in all cases, the way that the information is carried out is a gene is transcribed. That is the name of the copying mechanism that produces an RNA copy of a given gene. And then if it is in fact a protein-coding gene, that copy or messenger RNA will be utilized as the instructions to synthesize a specific protein that the cell needs or requires. Non-protein coding genes are also transcribed. The transcript can be a number of different types of RNA molecules that we'll hear about later, but those RNA molecules are not themselves instructions to synthesize a protein. So for non-protein coding genes, the flow of information stops at transcription after the production of the RNA molecule. So what I want to do first, before we completely jump into the mechanisms associated with molecular genetics in this lecture and the next two lectures, is go back and review a little bit of information about nucleic acids in general. Nucleic acids, as you should recall, are one of the categories of macromolecules. Macromolecules have a defining characteristic, that is, most macromolecules have a defining characteristic, and that they are polymers. Polymers, again, are long stretches of repeating units over and over and over again. So nucleic acids are in fact polymers. The individual monomeric unit is known as a nucleotide, and we're going to look at that structure in this particular slide in just a minute. But there are two categories of nucleic acids. There is DNA, which is short for deoxyribonucleic acid, and RNA, which is short for ribonucleic acid. In this figure, you can see the nucleotide structure for both a DNA nucleotide as well as an RNA nucleotide, and I'm going to highlight some of those differences here in just a minute. The one thing I want to mention, though, is that every single nucleotide will have a phosphate group, a 5-carbon sugar known as a pentose sugar, and a nitrogen-containing base. In the bottom left-hand side, what you're looking at is a nucleotide that would be utilized to synthesize RNA. And on the right-hand figure is a nucleotide that would be utilized to synthesize DNA. Structurally, they are very, very similar. Again, all nucleotides have at least one phosphate group, a 5-carbon sugar, and a nitrogen-containing base. For the RNA nucleotide in the bottom right, the first thing I want to focus on is the sugar. The sugar is a 5-carbon sugar known as ribose, and the sugar in DNA nucleotides is also a 5-carbon sugar known as deoxyribose. For ribose, the carbon that is in the number 2 position has a hydroxyl group associated with it, whereas in deoxyribose, that carbon is lacking in oxygen, hence the term deoxy, meaning without oxygen, in that number 2 carbon position. So again, the sugar for RNA nucleotides is known as ribose, and the sugar for DNA nucleotides is known as deoxyribose. Every single nucleotide will also have a nitrogen-containing base, and you can go back to the previous slide to review the position of that nitrogen-containing base. What I want to highlight with this particular slide is that for DNA nucleotides, there are 4 distinct bases that can be found. Adenine, abbreviated with an A, guanine, abbreviated with a G, cytosine, abbreviated with a C, and thymine, abbreviated with a T. In RNA, the bases are adenine, guanine, cytosine, and uracil. So that is a difference between DNA nucleotides and RNA nucleotides, and the first 3 bases can be the same in both categories. However, thymine is found exclusively in DNA nucleotides, and uracil is found exclusively in RNA nucleotides. This is a really nice slide because it highlights a number of characteristics of nucleotides that I just mentioned and a few additional things that I want to mention. At the bottom left, highlighted in the green, you can see the 2 distinct sugars that make up the DNA nucleotides or the RNA nucleotides, deoxyribose highlighted in blue and ribose highlighted in pink. So again, that clearly identifies the distinction between those 2 sugars in nucleotides for DNA or for RNA. The other thing that this particular figure also highlights is that for the bases, there are 2 broad categories of bases. There are pyrimidines, which are 6-membered rings, and there are purines, which are 6-membered rings fused to a 5-membered ring. The pyrimidines are cytosine, thymine, and uracil, and the purines are adenine and guanine. Now, the other thing that this particular slide highlights in the far right is that when we talk about DNA structure, DNA is a double-stranded molecule, and we'll highlight that and review it in the next few slides. But the interesting thing that allows for the double-stranded nature of the molecule to exist is the fact that the bases are able to interact with one another via hydrogen bonds. Adenine will always hydrogen bond with thymine, and thymine with adenine, and guanine will always hydrogen bond with cytosine, or cytosine with guanine, and that's shown in the far right figure in this particular slide. Before we move on, I want to talk a little bit about the elucidation of the structure of the DNA molecule. Many of you are hopefully familiar with the fact that Watson and Crick are credited with the discovery of the structure of the DNA molecule. In this particular figure, what you see on the left-hand side is the promo figure that they utilized when they first published their paper in 1953, where they explained the discovery of the structure of the DNA molecule. And then on the right-hand side, what you see is a picture that they recreated at the 50-year anniversary of that paper publication. The one thing I like to point out is that I always thought it strange when I was a student that here they are, these 2 famous scientists standing in front of a model. But it turns out that they actually worked with models extensively to try and elucidate the structure of the DNA molecule. Now, there's a lot of additional experimentation and other scientists that contributed to the work of their discovery, but one of the things that they did in fact do was work with these molecular models to help try and determine what the structure of the molecule was. I always like to go back and talk a little bit about the work of Rosalind Franklin. Rosalind Franklin was a female scientist. She had a PhD. She was an extremely well-studied and smart woman. She happened to work in the lab of a scientist known as Maurice Wilkins. Now, Rosalind Franklin was really good at a technique known as X-ray crystallography. In short, X-ray crystallography allows X-rays to pass through aligned molecules to look at and analyze the diffraction patterns that are formed of these molecules when the X-rays hit them. The picture that you see on the right is her classic figure that helped with the elucidation of the structure of DNA. Now, I know we look at that and we don't necessarily figure out or see how that shows a double helix molecule. But there's a lot of complex mathematical modeling that goes back with this diffraction pattern to go back and calculate and determine that the structure was in fact a double helix. Watson and Crick were given these figures, or these X-ray crystallography images, by Maurice Wilkins without Rosalind Franklin knowing. And so not only were Watson and Crick working with these models, they also had this data of Rosalind Franklin's to help them put the pieces of the puzzle together. Now, Watson and Crick and Maurice Wilkins won the Nobel Prize for elucidating the structure. Unfortunately, Rosalind Franklin did not get credit at the time. She had passed away from ovarian cancer. One of the downsides of working with X-rays back in the 1950s before we understood the problems that can cause to our DNA. Moving along, though, Watson and Crick established that there were in fact two strands of polynucleotides that come together to form the complex shape of the physical structure associated with DNA. So in this figure, what we're looking at is, in panel A, a single strand of DNA, which is referred to as a polynucleotide. It just means that it has nucleotide after nucleotide after nucleotide covalently linked to it. It is a more idealized version of a single-stranded molecule where the sugars are represented by the yellow squares. The blue represents the phosphates are represented by the yellow squares, the blue represents the sugar, and then you can see the bases individually labeled in this single-stranded polynucleotide. Now, in the structure of DNA that we find inside the cell, it is in fact two strands of polynucleotides that come together. The bases point inward towards one another, and like I highlighted on the previous slide, that hydrogen bonding that occurs between specific base pairs is what allows for the two strands to come together. The two strands interact and form this helical shape like you see in figure B. I typically like to include this figure because it really does simplify the way we look at DNA double helix. So if we work our way back from a double helix to the single nucleotide starting on the left, you have that classic helical shape, looks sort of like a spiral staircase. If you unwind that, it looks somewhat like a ladder, but then what you can see is a single strand represented in the next panel over. That simplified view is highlighted in a little more detail in that central figure where you can see, again, the phosphate, the sugar, and the base. And then that's highlighted even more so that you can see the nucleotide structure. So our lecture started at the right-hand side, sort of working at the nucleotide level and building a DNA molecule working towards the left. And then lastly, this particular slide has a really nice figure associated with it, where in panel A, you see the characteristic double helical shape. In panel B, you can see if we untwisted the double helix, sort of straightened it out, what you see is the two strands of single DNA coming together with the bases pointing inwards, and that hydrogen bonding is what's holding the two strands together. The other thing that's really nice about this particular figure in panel B is that it shows you the fact that DNA strands are what are referred to as being anti-parallel. It means that they are going in opposite directions of one another. If you look really closely, the polynucleotide that is on the left-hand side in panel B has one direction associated with it, and then your polynucleotide that you see on the right-hand side is basically in an inverted or upside down position. So those two strands are going in opposite directions of one another. That's not a mistake. That's exactly how they look, and this is referred to as being anti-parallel. Finally, in panel C, you can see the space-filling model of the double helical shape. Now, as I mentioned at the beginning of the lecture, there are several focal points of this particular lecture that I want to highlight. And so now we're transitioning into linking a little bit more of the information that we talked about with Mendelian genetics, more specifically with Mendelian genetics as well as with mitosis and meiosis. We talked a lot about chromosomes overall and chromosome movement. Now, what I want to do here is give you an understanding of how chromosomes might actually look in a typical eukaryotic cell. So I've established that DNA is in fact the genetic material. It contains all of the information that we need for a cell to be able to survive and function. But the DNA is not found in this naked double helical state in the nucleus of the cell. It is found in complex organization with proteins that are known as histones, and we're going to look at that in more detail on the next slide. But it is found in this complex organization of proteins known as histones, and this complex organization of the DNA with the histones eventually leads to what we know as a chromosome or the chromosome structure. So again, I just want to tie in this idea of DNA not necessarily being in this naked form in the nucleus, but in a more complex, condensed form that forms the chromosome structures, again, that we talked so much about for mitosis and meiosis. So this represents a more detailed view of the organization of the genome in the nucleus of the cell. Up at the very top of this particular figure, you see the classic double helical shape of DNA. Again, what I loosely refer to as naked DNA. It's just the DNA in its double helical shape. The first level of organization of the DNA in the nucleus of a eukaryotic cell is to associate that DNA with a group of proteins known as histones. Now, in this one step down figure that's labeled as beads on a string, this is exactly what that looks like, in that you have the DNA wrapped around the core of histone proteins twice, and then the DNA is wrapped around the next core of histones twice, and then the DNA is wrapped around the next core of histones twice, and this occurs over and over and over again. This is the first level of compaction of the DNA, an organization of the DNA. This allows for all of the DNA to fit into the nucleus of a cell in an organized fashion. These individual structures are referred to as nucleosomes. So a nucleosome is a core of histone proteins with the DNA wrapped around it twice. So in this figure, the second image down, you have 1, 2, 3 nucleosomes, one right after the other. In the cell, we would have hundreds and thousands of these nucleosomes linked one right after the other. And that's what you see in the next panel down. The next panel down is listed as the 30 nanometer fiber, and this 30 nanometer fiber is a condensation further of that structure that we just looked at. So in the structure we just looked at, there's a little bit of slack in the DNA between nucleosome to nucleosome. That is tightened up so that the nucleosomes are touching one another like you see in the 30 nanometer fiber. So we're further squishing or condensing the genetic material. Now, if you can envision taking that 30 nanometer fiber and looping it back and forth like you see in the next panel, that produces what's known as the 300 nanometer fiber. And this is, again, another way of condensing and organizing the material so that it's going to fit nicely into the chromosome itself, into the nucleus itself. Lastly, at the very bottom of the figure, you see this characteristic X-like structure. And I mentioned this several times in the previous sets of lectures, particularly when we discussed mitosis and meiosis, in that chromosomes don't look like this in the normal everyday life of the cell. It is when the chromosomes have been duplicated or replicated and condensed and entering into either mitosis or meiosis where we might see a structure like this. So that characteristic X-like structure is a completely condensed chromosome that is entered into either mitosis or meiosis. So the last focal point that I want to discuss for this lecture is yet another transition in this lecture, and that is to start discussing the process of DNA replication. So again, going back to mitosis and meiosis, one of the key defining features of entering into mitosis or meiosis is the copying of the genetic material, that replication process of all of the genetic material that happens in the S phase of interphase is crucial for mitosis and meiosis to occur. You have to have all of the genetic material copied in order for either one of those mechanisms to proceed. So what we're going to do now is look at the molecular level, how that replication process does in fact take place. What I like to start off with when I begin discussing replication is the concept that Watson and Crick actually published in a paper right after they published the structure of the DNA. They published a second paper that had no scientific data in it whatsoever. It was a speculation piece where they suggested how DNA replication occurs. In that paper, this quote has been extracted, and it says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism." That specific pairing that they were talking about is the base pairing that occurs specifically between C's and G's, or G's and C's, and the pairing that occurs between A's and T's, or T's and A's. That's referred to as the base pairing rule. It's also referred to as sequence complementarity. That is, if you know the sequence of one strand, because of the base pairing rule, the way that the nucleotides have to interact, the bases in the nucleotides have to interact, you automatically know what the other nucleotide is in the other strand. So what they proposed is a mechanism known as semi-conservative replication. And in this mechanism, what they proposed is that the two strands of DNA have to separate. And because of this base pairing rule or this sequence complementarity, if you know the sequence in one strand, you can fill in the opposite information because of the base pairing rule. And so with semi-conservative replication, what they proposed was again that you have the two strands of DNA separate. Each one then acts as a template for the synthesis of the missing information, and that occurs because of that specific base pairing that they had proposed. So with semi-conservative replication, you have a double-stranded DNA molecule at the beginning of the replication process. At the end of the replication process, you now have two double-stranded DNA molecules where one strand in each of the molecules represents the original parental or template DNA, represented by the red in this figure, and one strand represents the newly synthesized DNA, the DNA that was synthesized in the replication process. That's indicated by the green in this particular figure. One thing that I haven't reviewed in this particular lecture is something I'm going to ask you to go back and review on your own from our original discussion of DNA structure. Early on in the semester, we had our lecture that dealt with macromolecules, and then specifically I went back in and added a really short snippet of a lecture that talked about DNA polarity or DNA directionality, if you want to call that. So in that particular little mini lecture, I discussed extensively the difference between what's known as the 5' end of a polynucleotide versus the 3' end of a polynucleotide, or an individual nucleotide's 5' end and 3' end. So you absolutely should go back and review that because it's essential for understanding DNA replication. It's essential for understanding transcription, which is going to be our next lecture, and it's also essential for understanding protein synthesis or translation. So you have to understand that DNA polarity or directionality and where the 5' end and the 3' end reside in order to understand the next set of information. That being said, this is a slide that has an overview of DNA replication. In the top panel, you see a double-stranded DNA molecule in the dark blue. Obviously, in this case, the helix has been straightened out. It's not in the helical shape. The first step is to have the two strands separate from one another. Once the two strands separate, because of that base pairing rule, you automatically know how to fill in the missing information. And that missing information is being filled in the light blue. That's the newly synthesized DNA in this round of replication. At the very far right, you see two double-stranded DNA molecules. In each of those two double-stranded DNA molecules, you have a dark blue strand, which represents the original DNA all the way to the left, and then the light blue, which indicates the newly synthesized DNA that occurred in that round of replication. If you hone in on the second figure down at the bottom, what you can see is that there is a directionality for synthesis to occur. The 5' end of the incoming nucleotide is always going to attach to the 3' end of the growing chain. So you can see that in the left-hand side of that panel at the bottom. The nucleotides that are required to synthesize DNA and RNA are triphosphates. They have to have 3 phosphate groups covalently linked to them. What happens in the process of DNA replication is that the 2 end phosphates are removed and that single phosphate still remains in the nucleotide structure as it's added on to the growing chain. So again, DNA synthesis always occurs in the 5' to 3' direction. The 5' end of the incoming nucleotide attaches to the 3' end of the growing chain. Okay, so now let's look at the process of DNA replication in a little more detail. We're going to talk a bit about the enzymes that are involved in helping this replication process along. The first step in DNA replication is what's referred to as initiation, starting the process, getting it initiated. How does that happen? Well, Watson and Crick had proposed that the strands must separate, and in fact, that is exactly how DNA replication is initiated. There are enzymes that are known as helicases, and helicases are enzymes that can unwind or separate two strands of DNA. So helicases recognize a position in the DNA and begin to unwind the DNA. Once that DNA is unwound, what has to happen is proteins need to bind to those single strands of DNA to prevent them from closing back up again. There is a great amount of pressure to have those hydrogen bonds reform that hold the two strands together. So if we have the helicases coming in and unwinding or separating the two strands, there needs to be something that's going to prevent those two strands from closing back up again. And that is with the help of SSBPs. SSBP is short for single-stranded binding protein. These single-stranded binding proteins do exactly what their name implies. They are proteins that bind to single strands of DNA, and in this case, they prevent the closure of those two strands so that replication can proceed. In addition, there is an enzyme listed here as DNA gyrase, which is an older term. The more modern contemporary term is to refer to that enzyme as a topoisomerase. This is an enzyme that binds to the DNA, makes a cut in one of the strands to basically allow for a pivot or a swivel point. Because when you begin to unwind the DNA, the DNA downstream of that opening, also known as the replication bubble, begins to constrict, and that can cause breakage or damage of the DNA. So by having an enzyme that makes a nick or a cut in the DNA allows for a swivel point to ease or alleviate some of that tension. Now that we have the DNA opened up, we can begin the replication process. The next enzyme that comes into play is an enzyme known as primase. Primase is an enzyme that is known to be an RNA polymerase. That means it's an enzyme that synthesizes RNA. Interestingly, the enzyme that's going to do most of the work during the DNA replication process is an enzyme known as DNA polymerase, an enzyme that synthesizes DNA. But DNA polymerase lacks the ability to recognize a single-stranded DNA template and start adding in nucleotides. It has to have an attachment point to begin adding nucleotides to. Primase, which again is an RNA polymerase, doesn't have this same problem. It can recognize a single-stranded template and begin adding in RNA nucleotides. And that's exactly what it does. Recognizes a single-stranded template, synthesizes a short stretch of RNA that is referred to as the RNA primer. And then once this primer has been added, the DNA polymerase can jump on and start adding DNA nucleotides. And so that's sort of what you see in the center panel, with the pink strand being the DNA template that is being copied, the orange representing the RNA primer that is synthesized first, and then the blue representing the DNA that is synthesized once DNA polymerase can jump on. As I mentioned when we were discussing DNA structure, the two strands of DNA are anti-parallel. That means they're going in opposite directions of one another. This presents an interesting and unique problem for DNA replication, in that the enzymes that are involved in the replication process sit at a location known as the replication fork. And in this particular figure on the right-hand side, that is what you would see at the top panel, sort of at the Y junction. That's where the enzymes would all be. As the DNA unwinds, moving towards the right, DNA synthesis on that bottom strand can just continue without any problem. So if you just envision that bottom strand as we look down at a sequence of time steps in this figure, as long as the DNA is unwound, the enzymes keep going in that direction and keep synthesizing DNA. This is referred to as continuous DNA synthesis. It keeps going without any stopping, and this occurs on what's known as the leading strand of DNA. No problem there. The opposite strand, however, is going in the opposite orientation, so the synthesis needs to move away from the replication fork. This is a structural and space-constraining type of problem. The enzymes are moving towards the right in this figure, but synthesis is moving in the left in this figure. The solution for this is to have what's known as discontinuous DNA synthesis. DNA synthesis in short bursts, and this occurs on what's known as the lagging strand. These short bursts of DNA synthesis are known as Okazaki fragments, and they are named after the scientists who discovered that this is how DNA synthesis was occurring on what's known as the lagging strand. What this figure does is try to put together all of those pieces of the puzzle that we just talked about. So what we have is a number of enzymes that are playing a role in the DNA replication process. Helicases are involved in unwinding the DNA, and that has to occur throughout the entire DNA synthesis process. Single-stranded binding proteins have to bind to single-stranded DNA, prevent the closure of that DNA. This also occurs throughout the replication process. Topoisomerase is that enzyme that makes a single cut, allows a swivel or a pivot point to relieve tension that has been generated by unwinding the DNA. That also occurs throughout the entire process. We then move on to what we might consider the elongation process of replication, and that is where we have the need for the enzyme primase to synthesize the RNA primer. We have DNA synthesis occurring by an enzyme known as DNA polymerase, or DNA Pol, and in this figure is listed as DNA Pol 3. There are, in fact, a number of different types of DNA polymerases, and DNA polymerase 3 is the main workhorse. Its job is to do the synthesis of DNA during the replication process. Also listed on here is an enzyme known as the sliding clamp, and the sliding clamp binds the polymerase, the DNA polymerase, to the template to make sure that that polymerase stays on track, doesn't fall off, continues the DNA synthesis process. So the sliding clamp is a really important enzyme as well. You also see on this slide primer removal and replacement. So you have a situation where RNA primers are synthesized because the DNA polymerase cannot just jump on and start synthesizing DNA. But a DNA-RNA hybrid molecule is actually much less stable than a molecule that has DNA in both strands. So what has to happen is every single place that there is an RNA primer, it has to be removed and filled in with DNA. There is a special DNA polymerase that does this, and it is known as DNA Pol 1 or DNA polymerase 1. Lastly, there is an enzyme known as ligase, and ligase closes up the covalent linkage between Okazaki fragments, for example. And that is the DNA replication process in a nutshell.